Hello everyone, I'm Maryam, and today I'm excited to present MOMP, the matrix profile optimization for motif discovery. This algorithm was developed at a time series lab at UC Riverside under the supervision of Professor Eamonn. Motif discovery is a critical task in time series analysis, as it involves identifying conserved patterns or repeated behaviors within datasets. These motifs have applications across various fields, from genomics and healthcare to industrial monitoring and environmental studies. Existing methods for motif discovery include matrix profile, or I will call it MP from now on, which is efficient but constrained when applied to large-scale datasets, typically handling time series only up to a few million data points. Other methods rely on pruning calculations, but are often limited by time and space constraints. MOMP overcomes these challenges, enabling efficient pruning and scalability to much larger datasets. One of the main contributions of MOMP is the introduction of the first ever lower bound for MP. To explain this, let's begin with a geometric perspective. Imagine 2 points, R1 and B1, whose exact distance is known. Each point also has a nearest neighbor, R2 and B2. Instead of calculating the exact distance between R2 and B2, we apply the triangular inequality twice to estimate a lower bound for the distance. This approach is computationally efficient and still provides meaningful and precise approximation. The same idea works if we know a lower bound for R1 and B1 distance and not the exact distance. And it leads to a new lower bound, which is bigger but much faster to compute for the distance between R2 and B2. In the time series domain, this principle is applied through downsampling, and here's how it works. The downsampled time series T prime has a stepwise shape, as you see in this figure. The corners of the downsampled steps serve as anchor points. Neighboring points within each step forms cohorts. AMP is MP applied to T prime, which is a lower bound only to the anchor points. Using triangular inequality twice, we compute a lower bound for all data points in T called lower bound MP. By first calculating the MP on the downsampled data, we derive a fast approximation. Using the triangular inequality, we refine this into a tight lower bound. This lower bound is novel and allows us to prune irrelevant data points early in the computation, drastically reducing processing time. To the best of our knowledge, this is the first ever lower bound for MP. An essential feature of MOMP is its ability to balance the tightness of the lower bound with computational cost. Tighter bounds allow for more precise pruning, but require more computations, while looser bounds are faster to compute but less effective in pruning. This trade-off is controlled by the downsampling rate. A downsampling rate of, let's say, 1:1 generates an MP nearly identical to the original matrix profile. 1:1 is exactly identical, but it is computationally intensive. A high downsampling rate, let's say 1:16, reduces computational costs while yielding coarser bonds. MOMP progressively refines the downsampling rate in steps, optimizing efficiency while preserving accuracy. So now let's look at the iterative process of MOMP. MOMP combines downsampling with iterative pruning to efficiently identify motifs. Start with a high downsampling rate. For now, assume 1:16. Compute the lower bound MP and prune all data points above the best so far threshold. Half the downsampling rate and repeat the process on the remaining data points. In each step, we half the downsampling rate, and in the final step, compute the exact MP on the reduced dataset to identify motif. This method significantly reduces computational overhead while maintaining exact results. Now I would like to talk about key features of MOMP. MOMP offers several advantages over traditional methods. MOMP results are exact. It guarantees precise motif discovery. It doesn't require any user-defined parameters. just the time series data itself. MOMP processes datasets with hundreds of millions of data points, making it suitable for large-scale applications. It enables early termination of computations with meaningful intermediate results. Even in worst-case scenarios, where there are no motifs in the data or all the data points are motifs, the overhead is limited to just 33%. Also, MOMP is independent of the matrix profile algorithm. You can use any of the versions, CPU or GPU based, and it is independent of the lower bound that is used. MOMP has demonstrated its utility across a variety of real-world datasets. You can find a detailed explanation of the experiments along with more results on MOMP website. But for now, I would like to review some of the interesting results we got. We tested MOMP on so many real-world datasets, and here you can see 25 datasets. Although we know it's possible, but we didn't get negative speed up in any of the experiments we did. And on average, on these datasets, we got over 100 times the speed up. Some more experiments, I can point to industrial monitoring, where MOMP identified operational anomalies in secure water treatment systems, aiding in system reliability. And this experiment was the only one where quick motif could handle because of the short lengths of motif. And if we increase the lengths of the motif, quick motif would fail. Another experiment where we tested sleep apnea, MOMP analyzed oxygen saturation level, identifying motif related to desaturation and reoxygenation cycles. These findings were confirmed by medical specialists as clinically significant. In the wind turbine set, MOMP found an interesting motif, which includes a precursor dropout. If we zoom in to the motif that is found, this dropout is right before the step down, and this gives us a better view of how the system is working. In environmental studies, MOMP detected moments when birds transitioned from bleeding to flapping, providing insight into energy expenditure. We also use MOMP in genomic research. In chromosome 1 analysis with over 200 million points, MOMP identified large motifs in under 2 hours on just a regular desktop machine. The analysis of chromosome 1 with motifs exceeding 65,000 points achieved exceptional pruning rates. MOMP reduced the data to just 3% in the first steps and performed exact calculations on less than 1% of the remaining data points. This scalability highlights MOMP efficiency for large-scale datasets. To compare, a scan would have required over 5 years for the same task to do. We also continued this study by going over all other chromosomes with interesting observations, which I'm going to skip for now. Also in this study, we used early abandoning, which is a very important, interesting feature in MOMP. And finally, I would like to talk about a cross-species comparison we did. MOMP revealed conserved and inverted motifs between human and chimpanzee chromosomes, offering insights into evolutionary biology. MOMP was used to identify large-scale inversions between human and chimpanzee chromosomes, numbers 17 and 21. Inversions in chromosome 17 were confirmed through detailed comparisons, demonstrating MOMP's effectiveness in across time series motif analysis. Also, although each of these experiments took about 17 hours, but also we got very precise result even at the very initial step, right after about 20 to 40 minutes per experiment. To conclude, MOMP represents a significant advancement in time series motif discovery. By introducing the first ever lower bound for MP, it addresses the limitation of traditional methods and scales efficiently to large datasets. Its combination of efficiency, accuracy, scalability makes it a versatile tool for applications in genomic, healthcare, environmental monitoring, and more. MOMP can be used either by specialists in diverse domains or by downstream algorithms. I would like to thank you so much for your attention, and I'm happy to answer any questions.